Welcome back. Today we're going to be talking about cell differentiation and cancer. Right up the top, there is very clearly going to be, or content warning, there's going to be very clearly discussion of cancer today. So the cell cycle is a repeated series of events, right? It's literally in the name. There's the cell, cell cycle. And it's a series of events of the growth and division of the cell. The cell is always working towards division, even if it's not actively dividing. So there's 95% of a cell's time is spent in interphase. Interphase is literally anything that isn't mitosis. Mitosis is cell division, the actual process of division. We used to call this the resting phase of the cell because we didn't just remember old-timey scientists were looking at it and like, I don't know, the cell's sleeping, and then sometimes it splits in half. Whereas we know a lot more now. Things are get a lot more complicated the more we learn about them. We learn that there's loads of stuff that happens during the rest of the cell, but we call this interphase, and we call the rest of cell divisions mitosis. We can break interphase up into G1, S, and G2. The G's stand for growth, and the S stands for synthesis. It's when DNA is replicated. Now, the cell cycle, obviously that's a very simplified version. And again, the more we learn about it, we're going to learn a bit more about stuff, and we have to go into a bit more detail. Now, again, we still have broadly the same thing. We have interphase, which is G1, S, and G2. And we also have mitosis. Mitosis can be broken up into 5 different phases and is finished off by a process called cytokinesis. But that's it's prophase, prometaphase, metaphase, anaphase, and telophase. But these are not the focus of today's talk. Today, we're going to be talking about the process of how information is used, but we need to sort of know about the cell cycle in order to get through this. So critical to this is that this process is it's sort of like a clock, or it's sort of like if you've ever checked days off on a calendar as you've gone from day to day to day. That's sort of like how the cell cycle is controlled. It's sort of controlled by these things called cyclin-dependent kinases, which are enzymes that sort of put little phosphates on stuff. And it sort of ticks stuff off as it goes along. So the cell can always sort of reference, like, what time is it in the cell clock, right? Are we in interphase? Are we in S phase? Are we in G2? This is all determined by these cyclin-dependent kinases. And cyclin obviously has to do with the cell cycle. Now, the important thing here is that there are certain things called checkpoints. A checkpoint is it's represented by a stop sign, which is very apt in that cells can get to a certain point, and then they basically have to stop and check their work. They have to say, wait, hold on. Have we done everything correct up until this point? Now, there are 3 major checkpoints that are listed here, but there's loads and loads and loads of these checkpoints all along the way. The main 3 that we're going to be looking at here have to do with properly dividing, and the metaphase checkpoint is not as huge of a deal for us today, but the G1 and G2 checkpoint, these are basically like, we want to check the DNA before it's replicated, right? Because if your DNA's got a mutation in it and then you replicate it, well, now you're shit out of luck because you've got 2 mutations. So you don't want that. So you want to make sure that the DNA is all fine and dandy before you do DNA replication. Subsequently, once you've done that, the cell wants to check its work. The cell needs to make sure that everything's is done properly, and also that it's ready for mitosis to begin. Now, there are others along the way. There's one at the very beginning of G1 that says basically, if you don't, once you start G1, once the cell starts G1, it's committed to attempting cell division. Whether or not it dies along the way or what kind of cell division, that's not really here or there. But once the cell starts G1, it will go through to the end. Now, if the DNA is damaged, and that's what we're concerned about today, is DNA damage, apoptosis may occur. This figure originally said will. That's not true. Now, 2 things there. 1, it may occur. 2, what the hell is apoptosis? Apoptosis is it's a kind of cell death, and we'll get to that in a bit. But if the DNA is damaged, the cell may die. This is this will occur if the cell has damage that is to the point that the cell can't repair it. The cell will always try and repair its damage, the damaged DNA. But if it can't, then certainly in a multicellular organism, there are mechanisms in place where the cell says, oh, this is bad. Let's just shut this down. Something's gone terribly wrong. Shut it down. And for a cell, for an organism like humans, like us, we've got 37-something trillion cells. We lose a cell, no problem. Not a big deal. So this is a mechanism that sort of been, that evolution is selected for in order to protect the overall organism. I'm going to try and avoid sort of descriptions of, or sort of referencing it as like cell suicide, because I'm not super keen on that, but that is effectively what it is. The cell recognizes that it is a problem for the overall organism, right? Or there are mechanisms that recognize that it is that the DNA is damaged. You can see exactly why I try to avoid that, but that's the cell kills itself. The cell self-destructs is perhaps a better way to do it. Cells are like tiny little robots. All right, so if the damage is extensive, the apoptosis may occur. It will occur if the damage can't be fixed. And the same thing happens over here. If the DNA is replicated properly, apoptosis will occur if DNA is damaged and cannot be repaired. So this is all well and good, but it doesn't really get us to why we're talking about it in terms of cell differentiation, and what the hell is cell differentiation anyway. So this is the way a cell is supposed to work, right? And so this is sort of setting the stage, part one. We're going to jump now into sort of how another aspect of how multicellular organisms work, right? Multicellular organisms have apoptosis because they are multicellular. Obviously, single cell organisms are never going to do this because they only have one cell. They can't lose that cell. So branching from this, we're going to now go into a different aspect of multicellular organisms, which is that a weird thing, right? Because people like to talk about like, oh, well, it's such and such is in my DNA. I've got all this, like, oh, DNA determines everything. That, like, I think biology teachers of the past have sort of, in an attempt to like upsell and to try and convince students that like, oh, well, actually what we're learning is super important because DNA determines everything about you. And like, straight up, that's not true. That's not even everything you look like. There are many factors that are sort of determined after the fact, determined by just random chance. There's a great example of sort of like how many hairs are on each side of a fruit fly's wing, which is like, oh, well, this is basically random. It is almost never the same, right? And if it was, because of the way DNA works, because it's the symmetry, if it was genetically determined, it would always be the same. So there's, and like fingerprints, although fingerprints are a little dodgy. So basically all cells in an organism contain the same DNA. There's some weird exceptions like red blood cells. Red blood cells started out with all the same DNA, but then they had their nucleus taken out, and that's why they have that funny little button shape. So, but we can say all cells in an organism contain the same DNA, right? So from that, we should immediately be able to see that DNA must not be the be-all and end-all, because we are not made up of all entirely one kind of cell, right? We've got eye cells and leg cells, skin cells and leg cells. What the hell? We got neurons and kidney cells and liver cells and lung cells and epithelial cells and glands and all sorts of weird stuff that's in our body. We got loads of different types of cells. But they're all reading from the same book, right? They're all figuring out like, oh, well, they all have the same genetic code. So DNA clearly can't be the be-all and end-all. The answer to this is differentiation, which is enabled by gene expression. So differentiation is the process of cells becoming specialized. And perhaps this next slide should have been one ahead, but we'll deal with it. The central dogma of biology, which we'll remember, is that all information in the cell begins at DNA, is transcribed into RNA, and then translated into proteins. This can actually give us a somewhat incomplete idea or picture of how cells actually work, right? We think like, oh, well, therefore the DNA is the be-all and end-all. Again, the DNA is not the be-all and end-all. It's actually all going to come down to which genes are expressed, right? You could have the genes for wings, but you're not fucking flying, are you? So the determining which DNA is transcribed into RNA. This is the key right here. Right? This actually, which bits get transcribed. That's of critical importance. And this process in multicellular organisms, this is called differentiation. It's the process through which cells can become specialized. Not all cells within a eukaryotic organism even divide. If we go back here, you may have noticed G0. This is an extra phase that we didn't really talk about, right? G0. G0 is, it's terrible. Basically, not all your cells divide. Your skin cells don't divide. Or the cells on the outer layer of your skin they don't divide anymore. They're just sort of produced and then they live and then they're done, right? There are plenty of types of cells that don't divide. These cells they are in something called G0, which is a resting state. Now, cells whose daughter cells can become multiple different types of cells, right? So the cells that produce your skin cells can only become, they produce a skin cell and they produce a cell that makes a skin cell, right? They make a copy of themselves and they make a cell that is a skin cell. That's not a pluripotent cell. A pluripotent cell or a stem cell, an undifferentiated cell, is a cell that can become whose offspring can become multiple different types of cells. So a zygote, right? When during fertilization, when the egg captures the sperm, that cell, all of us originally were one single cell. That cell is definitionally a stem cell. It's a pluripotent cell. That cell had the capacity, or that cell's progeny had the capacity to become every single type of cell. But over time, little sections of that possibility got cut off, right? So we all started as a single cell, and then that divides and divides and divides and divides and divides. But then you have some layers of cells that eventually you get one that will become. Actually, hold on, let me find another slide to put in here. Okay, so I found it. So this is a slide so the fertilized egg, which is called a zygote. It eventually goes through multiple different rounds of cell division, and then eventually becomes a morula, a blastula, and then a gastrula. At this point, once this big ball of cells hollows out and then folds in on itself, at this point there's going to be sort of three layers of cells. There's going to be the endoderm, the ectoderm, the endoderm, and the mesoderm. At that point, cells that are on the ectoderm cannot become bone cells. They're cells they divide into, their progeny. Their descendants cannot become bone cells anymore, just like the endoderm cannot become skin cells. So this is sort of the first round of differentiation. This is the first time anything's cut off. So any sort of avenues or possibilities have cut off. So these cells all express, these cells all have the same genetic material. They all have the identical DNA. Like maybe there's a few odd mutations here and there because life is weird. But broadly, we can say they all have identical DNA. But what they do is they express they're neuron shaped, right? Because they're expressing genes that cause them to become neurons. So anytime anything happens inside of the cell, it is because of a protein. So proteins, that is the process of DNA, right? So genes are just, our definition of a gene is just a stretch of DNA that codes for a protein. The proteins are the things and we have multiple different classes of proteins. So we have structural proteins, the proteins that help the neuron have its shape and the epithelial cell have its shape are proteins. There are little scaffoldings inside of there that help them have that shape. The things that allow the surface receptors on the dendrites and the axons of the neuron, they are proteins. They are proteins that are involved in sending and receiving signals. Just the same way that epithelial cells or glandular cells your glands are a type of epithelial cell. They will secrete stuff, right? The things they secrete are done by proteins. It's done by enzymes. So what's actually happening here is that we have a whole book of, a whole encyclopedia of different proteins that the cell can express, and neurons only express the ones that have to do with being a neuron. And there's going to be some overlap, right? All of these cells need to be able to do glycolysis or something. So there may be commonalities here, but there are some cells, there are some genes that the neuron is expressing that the skin cell is not. And those cells would be neuron specific. So let's analogize this. Let's see if we can make this relatable. Imagine that your genome, right, that all the DNA in every single one of your cells, imagine that's like a catalog of Lego bricks. I love Lego bricks. It's a great analogy for just about anything I teach. So a catalog of Lego bricks is like a genome, but for Lego, right? You can make, you can order from Lego company, right, or whatever. A 2x2, a flat 2x2, a brick, plate 2x2, a brick 2x2, a plate 2x3, all of these different options, all these different shapes. You can order these, but not everything you want to build needs every single type of, not every Lego building needs every single type of Lego brick. Some, the pirate sets need specific pirate bricks, just like the skin cells need skin-specific proteins. Now, there are going to be general, right, probably every single Lego kit in existence probably has like the 1x1 brick or the 2x2 brick in there. But just because but it's the differences between which Lego building, which sculpture feels wrong, but which Lego thing needs which brick. Right, space things use space bricks. Pirate things use pirate bricks, and whatever other no idea. I would like to know more about Lego. Maybe when my kid's older. Right, so not every cell needs to produce or use every single kind of protein. I don't know what happened to the rest of this text. I swear to God I wrote it, but fix it. You didn't. Oh, there we go. Fixed it. You had no idea. You didn't. You noticed absolutely nothing. So not just in the same way that not every building needs, or like Lego creation needs every single Lego brick, not every cell needs to produce or use every possible protein. Now I want to return now to apoptosis, which we mentioned here. I even underlined it. Apoptosis is when a cell self-destructs. So we call this programmed cell death. This results from excessive damage from which a cell cannot recover. So normally cells have a certain shape, right? We just saw that our neurons have shaped like neuron cells are shaped like other neuron cells, and they have a different shape from skin cells, and different shape, columnar and squamous and cuboid cells. These are all different shapes of cell. So the first step, if a cell notices that there is something wrong, that would be a huge problem, and the cell might start growing out of control, or might rupture or die uncontrollably, it is better for the cell to self-destruct in a controlled manner rather than an uncontrolled manner. Now, the first step here is that the cell is going to round up and rounds itself off. It abandons the shape it has, and the nucleus starts to collapse in on itself. The chromatin is going to condense, the nucleus fragments into different bits, and then these little things called blebs form. This is called blebbing. It's a really exciting word to say, blebbing. But effectively what the cell does is it breaks itself up into little fun-sized, bite-sized chunks for the immune system. Then the phagocytes come along and they eat them up. This is the way things are supposed to happen if the cell is dying properly, if the cell is self-destructing properly. It's actually a good thing, right? So we might think of this being like, oh no, we want to protect, keep every cell is precious. We want to keep every cell. No, fuck that. Cells are dispensable, right? Cells that could cause a larger problem by hanging about should be destroyed by the body, right? This is this is like the non-ironic sort of greater good sort of use, right? So we're using the greater good sort of in a not shitty way. So cells, this is because if the cell were to just like die on its own, it would probably just burst open, and cells contain a whole bunch of stuff, right? They contain signals that they might want to send to other cells, but like don't want to send right now, right? So if they just burst open, release every single kind of signal at once, or nucleases, or things that eat up proteases, things that are going to eat up the proteins that are on the surface of other cells, it can cause so many problems. Uncontrolled cell death is actually, we have a name for it. It's called necrosis. Necrosis, it also causes an inflammatory response in surrounding cells. And inflammatory responses are problematic. They take energy. It's no good. So the body wants to avoid necrosis, and because of that, evolution has sort of has adopted this system, or ended up adopting the system of apoptosis for multicellular organisms. We can compare and contrast necrosis versus apoptosis here. Necrosis it tries to undergo fashions of it tries to undergo apoptosis. It doesn't actually work. The blebs eventually fuse, and the cell will eventually rupture. Now, apoptosis is a crucial tool for multicellular organisms. It does loads of different things. It's responsible for a bunch of different stuff. This is including things like, it says on screen, right? It's a major tool in protecting against cancer. But it can also do, it's also very important for other things, right? So there is space between your, when we were all a fetus, we had, between our fingers, we had web fingers, right? We had skin that ended up being between our fingers. And what ends up happening is that the cells in between our fingers ends up dying, right? And they go through apoptosis. So apoptosis is actually crucial for regular organismal development, in addition to protecting against things like cancer. But we're going to talk about it right now in the context of protecting against cancer. But that is not the only context in which apoptosis is used. So, cancer. Let's get into it. Cancer at its most basic level is just uncontrolled cell growth. It is cells that are growing over and over, or they're growing too fast. Cancer is the result of mutation as well. So cancer is uncontrolled cell growth, and it is the result of mutations. Now, there are three things that all cancers are different. So we tend to talk about cancers like, someone has breast cancer, someone has lung cancer, someone has skin cancer, but that's sort of a relative of when things were less understood, and that just sort of tells us where the cancer originated in, the tissue type, the tissue or organ type that it originated in. This is, and that can be helpful because different tissues express different genes, right? This is cell differentiation. This is why breast cancers behave a certain way, and they tend to behave different from colon cancers, for example. This is because breast cells or kidney cells or lung cells they have a different expression profile than the cells of the skin. So because of that, we see cells in, let's say, if you ever heard of like triple negative breast cancer, something that's estrogen receptors and HER2 receptors and the other receptor, which I can't escape in my head right now, but because breast cells express those proteins in higher abundance, they end up playing a larger role in breast cancers, as opposed to something like colorectal cancer, which ends up being something, usually it's associated with like basic excision repair or something. So we'll come back to this figure in a bit, but we'll start here. So what happens with cancers? Cancer is also the result of mutation. So new mutations arise, and one cell has the ability to start a tumor. Now, it's not even really a cancer yet. A tumor is just some uncontrolled growth, but as long as it stays in the place that it is, it's not really carcinogenic. It can be a problem, but it's not a cancer. A cancer is defined by it spreads beyond where it was supposed to be, or beyond the tissue type that it originated in. And now we have primary. So this cell can grow and grow and grow and grow, and it's because it's uncontrolled cell growth, right? The reason that you don't keep growing just more and more and more skin every day is because your cells are their growth is controlled, right? So only certain cells are dividing, and they're dividing at a certain rate. Cancer cells throw all that out the window, and they say, "Fuck it, let's just go, let's divide," and that's why we get the way tumors are lumps of tissue. This is because it's the result of uncontrolled cell growth. Now this is the primary tumor. And now these cells are going to accumulate different mutations over time. And so the cell originally, the tumor originally, the type of cell that it was derived from, right? Let's say it's a skin cell. Skin cells are set up to survive in a very particular environment. They're set up to be exposed to the air. Like neurons, your neurons really don't want to be exposed to the air. They die. So the skin cells tend to exist, or tumors tend to exist in the type of tissue that they started at. But as they acquire more mutations, right, because, like I said, not every cancer is the same, but in fact, not every every cancer is not every cancer is the same, but in fact, like every tumor is different because they are the result of random mutations. It's not some sort of defined mutation. It's every single cell is just acquiring random ass mutations, right? Mutation here, mutation there, and a lot of them don't do anything, but sometimes, right? Sometimes it can cause the cell to be able to behave incorrectly. And we are all acquiring mutations all the time. This is why your chance to get cancer increases as you age, because you've just had more time. If you acquire one new mutation every week, most of those mutations don't really do anything. We've got loads of DNA that isn't really involved in genes, that doesn't code for genes. But eventually, if you keep hitting that, right, it's like losing the shit lottery, right? Eventually, if you keep as time goes on and that rate of mutation remains constant, or as long as it's still there, as long as there is a rate of mutation, your chances of acquiring cancer increase over time. So each one of these cells, and so not even is every single tumor different, right? Because they're all unique. But the cells, not even within a tumor, every single one of these cells is acquiring their own random mutations. It's not like once this cell here gets a mutation, then it spreads to every other cell. It's just different cells are getting different mutations all the time. So not even every single cell within a tumor is the same. This is part of what makes cancer so insidious. So the cell acquired some mutations that allowed it to grow out of control, but as that rate of mutation, and within cancers, the rate of mutation is elevated versus the other cells. So over time, the cell may begin to, and these cells are adapted to, or are designed to live in the type of environment that they are, right? Again, neurons wouldn't be able to live out here, but that also means that these cells can't really grow inwards because this is a different environment than it was up here, right? The skin cells are supposed to be exposed to the air. They're not supposed to be in here. It's a different pH condition. The immune system looks at things differently in there, right? There's all sorts of reasons that the cell wouldn't be able to exist in here, but if you have enough time and enough random mutations, they will start to grow inwards, right? And we would say, if you have the pathologist that they were looking at this, they would say the tumor, this tumor has good margins, this tumor has poor margins, because this is sort of growing inwards, and once it grows inwards, it becomes less of a well-defined lump, and it can sort of grow down, sort of like roots of a tree. And then over time, that will eventually sort of find a lymphatic vessel or a blood vessel, and then you can have cells flowing throughout, cancer cells flowing throughout the body, and eventually they will land somewhere, or they will get mutations. They will keep trying, keep doing mutations, and bunch of cells will land somewhere, and they will just die because they don't have the right conditions, or they don't have the right mutations to live in some other environment. But eventually, the cells will sort of find another location that they've acquired mutations to be able to live in, and they will spread to other parts of the body. This is called metastasis. So I said all cancers are different, right? We'd say, even within one type of, even within something like breast cancer, which is something I did some research on, right? So even within breast cancer, there are different types of breast cancer. Not aromatase inhibitors or tamoxifen is not going to be effective against all types of breast cancer. So because of this, because all cancers are different, it's very, very difficult to treat. And it's also because it's your own body's cells that the immune system is kind of shit out of luck, right? The immune system is woefully inequipped to attack its own cells. And even though all cancers are different, there are the process of carcinogenesis of a cell becoming cancerous. Because there are some similarities here, we can say there are some broad similarities between all cancers. And again, there's an asterisk here. There's like a few here and there that are weird and don't actually fall into this. But broadly, for our level, this is a very good set of rules. So there are three things that all cancers have in common. First is that and these are not in any order. These steps, we could say these are three steps that cells have to pass through in order to become, in order for the cell to be cancerous, to become cancerous. But these don't have to happen in any order, and they don't even have to be one change. It can be a little bit of a change here, and a little bit of impairment of DNA machinery, and a little bit of impairment of something else, right? A little bit of impairment of turning off apoptotic pathways. Maybe they're just doesn't work quite so well, and then there's enough time for some other stuff to change anyway. So this is fluid. So all cancers, we tend to when we classify cancers genetically, we tend to associate them with some sort of DNA repair mechanism. So like colorectal cancers are often associated with something called microsatellite instability or base excision repair, whereas breast cancers can be associated with double-strand break repair because that's what the BRCA gene does. There are different types of cancers associated, but the thing here is that all of these mutations that occur, they are occurring ideally in a ideal situation, they would be fixed by the DNA repair machinery. But if that DNA repair machinery isn't working quite so well, then it can allow those cells that mutation to hang about, and then when the cell divides, now you've got 2 cells with the mutation, then you've got 4, then you've got 8, you've got 16, 32, 64, and eventually you get a lot of cells. That's what exponential growth is like. The next thing is that even if in a healthy cell, even if there were some DNA repair that wasn't getting fixed, the cell might notice it and be like, whoa, 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 whoa. Let's just shut this down. Let's self-destruct and protect the entire organism. So cancer also turns off apoptotic pathways. It prevents the signals from being sent. It turns off the enzymes that are involved with initiating apoptosis. Through whatever means, this cancer cells have turned off or impaired or shut down apoptotic pathways, and they have also turned off the cell's ability to check for them, right? So disabling cell cycle checkpoints allows the cell to undergo uncontrolled cell growth. So by disabling cell cycle checkpoints, it prevents the cell from being able to check for for DNA damage, or to even, if the checkpoints are off, then you can't initiate apoptosis. And these are 3 things that sort of all cancers have in common, and we have treatments that sort of that try to exploit these. So there are 3 main types of cancer treatment. There's surgery, radiation, and chemotherapy. These are there's never one single thing one single option is never correct in all situations. And again, I should always reiterate, this isn't medical advice. Please see a doctor for any medical decisions. But we're going to look at surgery. Surgery is a great option here in the early stages when it's a solid tumor. But once the cell starts to invade into surrounding tissues, right? Once those margins don't get so good, right? If you were to cut this out, if you ever tried to dig up a tree, right? If you wanted to get rid of a tree, let's say I've got a tree in my backyard I don't like. If I wanted to get rid of that tree, but I didn't really care about anything underground, and I just wanted to cut that tree, done. Very easy. Now, if the tree only existed above ground like this, right? If the roots never existed, trees would be really easy to get rid of. Surgery is an excellent option in early stages of cancer, when the tumor hasn't started to invade into surrounding tissues, particularly inwards, and particularly when it hasn't metastasized. Once it's metastasized, surgery becomes not really a very effective option because you're just cutting out a large group of cells, but there's still cells circulating in the body. It's very unfortunate. So if you were to try and remove this tree and all the roots, right? Remember, because of the way the cancer works, if you want to fully eradicate all the cancer cells, you need to get every single root. You need to get every single one of these roots. If you leave any, the cancer could come back. So once the cancer starts to invade into surrounding tissues, that becomes a very dodgy prospect because you may have to remove a large chunk, and let's say it's, in some sort of essential organ, like your lungs or something, right? Or your heart. You can't remove a large chunk of the heart and expect it to still work. This is why it's very important to catch cancers early. So that's surgery. Next up is radiation and chemotherapy. There are many types of chemotherapy, but generally, and along with radiation, they exploit the fact that cancer cells divide faster than normal cells, right? Because cancer cells have turned off the cell cycle checkpoints. They've turned off apoptosis, right? So they're just churning through. They're just dividing. So if we can muck about, if we can mess with the actual cell division process, or the DNA replication process specifically, and that's what radiation does, that's what a lot of chemotherapeutic agents do, is they just induce DNA damage, right? And the normal cell would be like, whoa, let's wait up. Hold up, we got to fix these because we have our cell cycle checkpoints intact. We need to fix these before we keep going. Whereas the cancer cell is like, fuck it, full steam ahead, let's just go. And that's not so, and so the idea is to try and, once you get enough DNA damage, the cell just won't be able to live anymore. So it just won't be able to function. So the idea is to cause the mechanisms of replicating the DNA with certain types of DNA damage that chemotherapy and radiation induce. It can actually really mess with the machinery of DNA replication. So this is what, so radiation and chemotherapy, these are things that will induce DNA damage. They're not things that you would want to be exposed to, right? It's sometimes it seems weird when people are like, oh, radiation causes cancer, but also we use it to treat cancer. And like, yes, that's true. But ideally, you would just avoid radiation altogether, but it is more lethal against cancer cells than it is against regular cells, and that's the big trick, right? It's actually shockingly easy to kill regular cells. Bleach works wonders. Cancer cells kills the shit out of bleach. Fire, also great. The big problem in cancer therapy, the big problem, is not hurting the healthy cells. You want to avoid hurting the healthy cells. That's the real sticking point. So right now, and there's a lot of cool stuff that's coming through the pipeline right now. I don't really have, this isn't the best venue to talk about something like synthetic lethality, but there are a lot of cool stuff that's happening in chemotherapeutics. Also, there's no secret cancer cure that companies are sitting on. Just the business model of the way pharmaceutical companies work doesn't allow for that. Right? If they had that, they would become the biggest company in the world, and corporations are inherently short-sighted. They just want growth this quarter, right? They want earnings immediately. If they had the cure for cancer, they would become the #1 company in the world, and there's no reason to sit on that. Also, if they were sitting on it, if I had discovered the cure for cancer and a company said, maybe we just don't sit. No, that's history books for me, right? Like, that is Nobel Prize at a minimum. And, right, so there's a lot of, I understand the idea as to why people aren't trusting in that, and I empathize with that, but in this case, I see no reason that there would be my professional opinion is that there's no reason that there would be a secret cancer cure that companies are just withholding from us. I'm going to give some cancer stats now. This is my one plea for, please don't smoke. So 1 in 7 people have cancer, some form of cancer in their lifetimes. 1 in 8 women will get breast cancer. Lung cancer is the leading cause of cancer death in the US, of which total cancer deaths is about 500,000 deaths annually, every single year. And lung cancer, we have data that shows when people started to smoke more, 20 years later, people started to get a lot more lung cancer. It is, that said, I understand that scared straight approaches don't work. This is why DARE as a program failed. But just, I don't know. This is my one plea, and then I'm done with it. So that's kind of a downer. So here's some pictures of my dogs. This was Zonda. This is Zoe. They're very good dogs, and I hope everyone has a good day. Thanks for tuning into the lecture on cancer and cell differentiation. If you have any questions, go ahead and shoot me a message. Thanks.